Hello, everyone. Good morning, good afternoon to all of our esteemed and attendees joining us from different countries and of the world. I am Pinar Sengul. You can call me Pi for ease of pronunciation. And it's my absolute honor to welcome you all to the International Live Conference on Drug Discovery for Brain Health, Mental Well-being, and Neurological Disorders, organized by the OLCIAS Live Conferences. Before we begin, let me take a moment to thank our incredible speakers, distinguished guests, and of course, all of you who have taken the time to be part of this insightful discussion of the drug discovery and neurological health. This conference is not just an event, it's a platform where we bring together some of the brightest minds to explore groundbreaking research and explore innovative solutions that can transform the future of neuroscience and mental health well-being. The theme of today's conference, Innovating the Future of Drug Discovery, Emerging Technologies and Novel Approaches, aims to shed light on the latest advancements in pharmaceuticals and therapeutics that can significantly impact brain health, mental well-being, and neurological disorders. From exploring novel drug therapies to understanding their applications in neurosurgery and psychiatry, we have an insightful lineup of discussions ahead. So I would like to introduce myself first. So I am Pinar Sengul. I'm a neuroscientist, and I am working with multiple sclerosis. And I also do some couples therapies and family therapies as well. I will also make a speech today on vegan and ethical future of the science. So I will be happy if you will take part of this today. So I will be the last speaker. So we have Dr. HABCHI Nawel today. She is chief editor of University of Victoria Journal, and she's a neurosurgeon. She's a very esteemed scientist. And we have one more speaker, which I will introduce now. Is she here? Nayana Ravindra Jawale, I guess, right? She's gonna make a speech today? Am I correct? Yes. Okay. Hello, Nayana. Are you here?
Hello, Dr. Pinar.
Hello, how are you?
I'm great. What about you?
I'm fine. Are you ready to make your speech?
Yeah, I can start.
Okay, is there any way that you can open your camera or not?
Yes, I'll open. Yeah.
Okay, great. Do you want to make a short introduction about yourself first, please?
Yeah, I'll do that. I'll just
Okay.
open my camera. Hello. Hi.
Hi. Thank you for opening your camera. That's very nice of you.
Yeah. And thank you so much for this invitation and accepting this one. I want to introduce myself. I'm a junior research fellow at MS Rama University, Applied Sciences in Bangalore, India. So, and I'm currently working on two approaches that I wanted to talk about it today. So it is basically I'm working on Alzheimer disease, but through the gut-brain axis. So
Wow.
that's, yeah. So like I'm exploring that through computational uh, chemistry, as well as the in vitro and in vivo studies and formulation as well. So that's about me.
Okay. So the gut-brain axis is a very important one in neuroscience. So,
Yes, definitely.
yeah, I'm so happy that you're doing that. Also, the intestines are called the second brain because
Mm,
they have so many nerve endings, as well as
yeah.
serotonin is also reproduced in the gut as well, right?
Yes,
There's
yes.
serotonin, just like the brain.
Mm, yeah. So can I start with the
So
presentation?
okay, you finished your master's, correct?
Yeah, I have finished my master's and I've finished my like 1.5 year of PhD also. Like still I'm in the second year PhD right now.
Okay, that is great. Okay.
So let's start with it, I guess. Can we start with the presentation?
Yes, you can.
Yeah. I'll just share my this one. Is my screen visible?
Yes.
Yeah. Okay. So I'll start with it. 1 second. Okay. So I'll start with my presentation. So I'm very delighted to present my research work, that is computational, and it is a part of my research work, that is computational exploration of symbiotic induced gut brain modulation in Alzheimer disease, a network pharmacology and molecular docking studies. So let's begin with it. As we all know, Alzheimer disease is neurodegenerative disorder and which is characterized by the cognitive decline, amyloid beta plaque, and neuroinflammation. And emerging evidence is suggesting that gut microbiota modulation via the symbiotic or probiotic techniques can impact on the neuroinflammation and neurotransmitter as well, and it will regulate the gut brain axis. So we thought of going into this deeper. So basically, in Alzheimer disease, it has been seen that gut dysbiosis happens. So what is gut dysbiosis? It is an imbalance of gut microbiota, or the death of good gut microbiota, and that leads to the neuroinflammation. So we thought of creating, or we thought of formulating something which can act on a gut and give results on the treatment of Alzheimer disease. So we have introduced this with the two plants, that is Brahmi, that is nothing but Bacopa monari, and Ashwagandha, and some sort of prebiotics which are already there, and also gut microbiota. Like we will specifically work on gut microbiota which are producing the short chain fatty acid, example butyrate, uh, and acetate, like that. So indirectly or directly, those will affect, or those will promote the neuroprotection. So this is the approach. So to carry out this study, we have chosen the two approaches. First is network pharmacology approach, and second is molecular docking approach. We have further more approaches, but still, like, we are, this is an ongoing project. So we are at the molecular docking approach here. So the first methodology of it is identification of the main active constituents of Bacopa monari, that is Brahmi, and Vitania somnifera, that is Ashwagandha plant. So how we have identified the active constituent of it? So we have extracted by taking the roots of leaves of Bacopa monari and roots of Vitina somnifera. After extracting, we have gone for analysis through the HPLC and LCMS technique. So from that we will get to know. Second is finding the network pharmacology network, like protein protein interaction, then which are the genes are involved in this and all. And third one will be, we'll go for the molecular docking technique, that is to check the binding affinity for this. And here we have used apps, or you can say tools, for the network pharmacology, which includes stitch database, gene card database. So these databases are like well known for the variety of genes. And for gut microbiota genes, we have chosen the gut m gene database.
Nayana,
After,
sorry to interrupt you, but are you moving the slides? Because we just see your title still.
yeah, I am moving the slides, is it not visible?
Yes, we are still just seeing computation exploration of symbiotic in the gut
Oh,
stream. Yes, now can you
can you
just
see now?
like presentation from slideshow?
Yeah, I
Slide,
was presenting
just
from the slide.
click
Okay,
on slideshow above. Can you see that?
yeah.
Near animation and review. Yeah, go down a little bit, down and right. Yeah, animations, then slideshow. Yes, slideshow there. Just near animations on the right. Yes, great. And then from beginning.
Can you see that? I was doing the same thing. Is it moving?
Oh, yeah, can you move? Now nothing is coming up, so that's the problem. Okay, your slideshow is not.
Can you see this right now?
Yes.
Okay,
Okay.
okay. I'm so sorry for this.
That's okay. No worries.
Okay, this was my second slide for the
Hmm.
introduction about the Alzheimer disease. Then, what was the main problem? That was the gut dysbiosis and leading to the neuroinflammation. Then the symbiotic approach. This was the hypothesis. And then I'll come towards the methodology. So these are all tools we have used. I'll explain this in detail in the next slides. So first one, identification of active constituents. So how we have identified? We have used two techniques. First one LCMS and one more is HPLC. So through this method we have got to know what and all bioactive compounds are present in it and in what amount. So from this report, we have analyzed that we have in Bacopa monari, we have bacoside A3, bacopaside 2, then bacopa saponins, bacosides, like different type of. So, and these are the two plants active ingredients or active components we have got from the LCMS and HPLC report. Then we'll come towards the network pharmacology. So now in network pharmacology, what happens? Basically, it is a analysis where we identify the key proteins or key genes which are mainly involved, and also the pathways. So here, so genes associated with the Alzheimer disease we collected from the gene card database, and genes which are associated with the gut microbiota are carried out or are taken from the gut M gene database. So total for Alzheimer disease genes we have got 15,506 genes, and for gut microbiota we got 117 genes. So to check how what and all the compatibility, or we can say how many genes are common with our extract genes. So what we did is we got the genes for this active constituents also. So gene associated with the active constituents were collected from the stitch database, that, and from that we got total number of genes. So when we want to identify the common targets, which are the common targets are there between the Alzheimer disease and active constituents, as well as gut microbiota and active constituents. So we need to carry out one Venn diagram analysis. So that we have carried out using the intervenn Venn diagram analysis. So from Venn diagram, I have displayed here two Venn diagrams. One is separated for the Alzheimer disease plus active constituent of genes, where I have found that from the 15,000 genes, found that 173 genes targets are common between the Alzheimer disease and active genes, active constituents genes. Similarly, in the right side, I have got around 8 common targets between gut gene microbiota and active constituent genes. So we have some connection through this, we can tell. And then when we move forward for the network pharmacology, there first we need to check the protein protein interactions. And to check that protein protein interactions, we have incorporated the common genes into the app that called cytoscape string app. So after incorporating of the common genes, we get to know the network. And as you see in the diagram below, the darkest color gene is is more, it has a more nodes, and it is connected to the more number of genes, like those all are interconnected. So the top one genes which have 25-degree layout, that is HSP 90AB1, and these are the total 15 common genes I have taken from the data, whatever the results I have got. So after this, common, after finding the common gist interactions, we need to go for the functional automation of high degree genes. So high degree genes we incorporate into this. So why we go for this co and cake pathway like analysis, what is the importance? So the importance is it helps to understand the biological function and disease mechanism, and also it identifies the molecular pathway affected in it. So in our case, it is Alzheimer disease and the gut microbiota interactions. So through this co and cake pathway, we will understand what and all the pathways which are associated, which are genes are involved in both the cases. And so from these pathway we can move forward for the further studies. So this is only of the Alzheimer disease thing. I have got around list 5 top target. I have got total 5 pathways which I have like selected and which have the more degree. So those I have selected through this for the Alzheimer disease. And through this we get to know that which genes are actually acting more on it. So these are the top 8 genes we got for this. After that, we will select this gene. These are the Alzheimer disease genes which are acting more. So if we block one gene also, it will ultimately block the others because it is interconnected. And the first node in the pharmacology in the network pharmacology indicates that if first node we are blocking, then ultimately it will block the other nodes which it is connected to. So these are the 8 genes which I have taken, and I'll move forward with these genes to do the molecular docking studies. Similarly, similarly, we have done for the gut microbiota and active constituent genes also. So from these, I have got this 5 genes common, and you can see that we have this MAPKI gene in both the things. It is common. So that means whatever the active ingredients I have selected, active constituents I have selected, those will act on the gene through the gut as well as to the human body, like the directly to the brain also in Alzheimer disease. So let's move forward for the molecular docking studies. So those 8 genes from the Alzheimer disease and from so 5 genes from the gut microbiota. I have done the molecular docking studies, and from that, the top one which I have got the top results, that one protein I have selected, and I'll go for the further activities. So here you can see the docking results. We have around, like, I have got around every all the active ingredients are showing around more than 7 binding affinity of this docking score. So, and the minus is the highly minus negative value, if we get that will be the good. And here in bacopa side 2, I've got the highest, that is minus 11.8. Similarly, if we go for the gut genes, and this protein which I have got from the gut genes, so with that also it is almost, I've got a good value of minus 11.6. And even the key interactions are showing that they have, strong binding affinity. So this was about the network pharmacology and molecular docking studies. So, what I want to summarize and conclude through this, like, combination of medicinal plant genomics, network pharmacology, and the microbiota research can lead to some novel therapeutic strategy for the Alzheimer disease. And advanced bioinformatics tools enables the gene disease drug target mapping for effective drug discovery. So this was not the complete project because it is ongoing. So it was just a, you know, a a short vision of my project. But yeah, the future research which I'm gonna carry out is I'm gonna take those complexes and go for the molecular dynamic studies. In molecular dynamic studies, I'll get to know about the stability of protein as well as my active constituent stability. So, how much time it is binding and how much time it is stable in the protein. So, after doing that and after getting the result, we'll be selecting the specific type of active ingredient plus my gut microbiota. So we have selected some four gut microbiota through these. So those are fecally bacteria, lactobacillus plantarum, Akkermansia mucifila, and Butyricoccus. So these four will be going with the compatibility study first to check whether those both are compatible or not. Because before doing the formulation, we should go for compatibility studies, because later we will not get to know if it is compatible or not. So first, doing the pre-checking of everything, we'll move forward for the symbiotic formulation, that is probiotics means gut microbiota, and prebiotic where my brahmi and ashwagandha's extract will act as a prebiotic. So both after combination will make as a symbiotic. After doing that formulation, we'll go for characterization of it, whether it is, stability, this thing and all. And after that, in vitro and in vivo studies, and in vivo studies will be carrying out using the vista rat model and zebrafish model. So this is about the project. And I'm very thankful for my supervisor, Dr. Suma Bibi. She is associate professor in the Department of Pharmaceutical Chemistry, Faculty of Pharmacy, MS Rama University of Applied Sciences, Bangalore, India. That she has helped me throughout this project, and it was a great support. Because of her, I have done this work, and because without her support, I can't do anything. So it was about her. So yeah, I would like to ask questions.
Yes, thank you very much, Nayana.
Thank
That
you
was a
very
great
much.
slide and great research.
Thank
I
you.
have two questions to you, which I typed down. The first one is like, from an engineering viewpoint, what are the biggest obstacles? Startup face in moving from prototype to clinical use. And the second one will be, what ethical or regulatory hurdles do you believe are underestimated by founders in the health tech space. You can start as much, whichever you like more.
Yeah, yeah, I'll just check. I'll just read the questions once. Is it in a chat box?
Yes, it's in the chat box, but I can send again.
Yeah, there is a third question that is like, based on your molecular docking studies, what specific advantage do
Mm.
prebiotic derived metabolites like butyrate offers in enzyme? Yeah. So I wanted to tell like, based on the molecular docking study, we will get to know because there is a specific protein which is which is butyrate coenzyme transferase protein. So it will
Mm.
be in the gut microbiota, and if we influence that protein, it act as an enzyme and it will convert acetate into the butyrate. So that is that protein if we are influencing, and if we are getting the good binding affinity, definitely it shows that it will produce the butyrate because it is about the protein. So if you are taking that protein and if you are getting the good results, we can ensure that we have got a butyrate producing effect. And if you want to check that, you can go from the in vitro activities as well.
Okay, thank you very much.
Yeah,
Can you ask for the other question, please, as well?
yeah. Yeah, there is a first question. Given in the complexity of gut brain axis in the Alzheimer disease, what challenges do you face in translating the computational prediction into the effective clinical applications? So yeah, definitely we 100% we can't rely on the computational system because it is just a prediction. But yeah, but it gives a very close prediction that we can minimize the other efforts and we can utilize that only in our future, like further research work. So whatever the result I have got from that, only like we are gonna use the in vitro and in vivo techniques. And after that, when we compare both in vitro and in vivo results with the computational results, that comparison shows how much similarity is there or how much we can rely on it. So yeah, definitely we just can't 100% rely on it, but yeah, we can take it as a prediction and we can minimize the other efforts of it. Like by taking, we could have taken any gene, right? But through this computational approach, we can get to know which pathway and which gene it is acting on, so we will be specific in our action. Yeah.
Okay, thank you. And my question was,
Yeah.
what ethical problems do you think arise in this research that you have, that you're
Which
doing,
one?
or the other people?
Can
Even
you repeat once again?
your, it might not be just you, but it can be other researchers
Yeah,
that
yeah.
are in the field. What do you think? That where is the boundary that we cross the ethics part, and we are?
Yeah, yeah,
I just want your viewpoint on this.
yeah.
Um,
As
ethical, yes.
yeah, as I mentioned, we are gonna do the in vitro studies also. So, and in
Mhm.
vivo studies, definitely we are gonna need animals. We are gonna need vista rats, plus we are gonna need the zebrafish model. So there the ethical committee plays a role. Like we need to explain them clearly what and all the parameters we are gonna use, what will be the toxicity studies and all. So for this also, when we go for the toxicity studies, before that what we do, we will go in silico toxicity studies first. So we will get the idea of, we'll get the results, and then if it is safer, then we can proceed with it. So for that ethical this thing and all, we need to present some computational proofs as well.
Yeah, okay, I get that. You're at least considering that there is some boundary being crossed in
Yeah,
animal
definitely there,
research.
there is a boundary. Yeah.
Yes, that's really good. That's really good thinking, because we tend to just repress that feeling, the conscience we have when we are doing research. for humans and maybe hurting animals on
Yes.
the side. And ethical considerations are, I think, a must characteristics of researchers.
Yeah, and
And
those are, I think those are really important.
yeah, I believe as well.
Yeah.
So yeah, I'm glad to hear that you're considering in vitro as well as in silico with the
Yes.
advanced technology that offers us many alternatives right now.
Yeah,
And
because
it also
blindly, if we
sounds
are going, yeah,
more
if
precise.
yeah, we will get more precise data with the in silico and this all in vitro.
Obviously.
That blindly, if
Obviously.
we go, we just can't do it on animals because we can't do it with them.
Yeah.
Ethical consideration we should follow.
Yes. Very
Yeah,
nice. Thank you very much, Nayana
yeah,
Ravindra. Is
yeah.
there anything else you would like to add? Nayana, is there anything else you'd like to add before I move on
Uh,
to the next speaker?
no, no, nothing.
Okay, great. Our next speaker is Dr.
Thank you
HABCHI
so
Nawel.
much.
Yeah, you're welcome. You can stay, and after Dr. HABCHI Nawel, I'll make a speech on ethical approaches in science. So
Yeah, yeah.
that might interest you
Yes,
instead.
definitely. See
Yeah,
you. Yeah.
yeah. Waiting for your questions as well. So you can stop sharing as well, Nayana. Can you also take off your slide? Yeah, thank you. Our next speaker is our esteemed speaker, Dr. HABCHI Nawel. She mostly presents her cases and her reports in OLCIAS live conferences. She's also in the team of OLCIAS, and she has many positions in scientific journals around the world. Very, very glad to know her in person as well. She's a neurosurgeon. She works in the neurosurgery department in France Fanon University Hospital in Blida, Algeria, but she's also connected to London, University of Victoria as well. Today, she's going to speak about future medicine, startups, and innovative applications. I will just briefly tell you about her abstract as well. She will be talking about the medical startups and then dive into the world of medical applications, the driving force behind healthcare innovation. She will talk about ChronoLab. A groundbreaking application will be highlighted as a model showcasing how technology enhances diagnosis, patient monitoring, and care optimization. At the crossroads of innovation and entrepreneurship, medical startups are shaping the future of healthcare today. Don't miss the deep dive into the future of medicine. I'm very proud to present this, as this is also very related with my research topic, presentation topic that I'm going to do after her. That medical startups with innovative technologies, we can finish animal experiments one day. Let's stick together and let's hear Dr. HABCHI Nawel's recording.
Hello everyone. I am delighted to welcome today to this exciting webinar on the future of medicine, startups and innovative application. As you know, the medicine is evolving at a rapid pace, driven by a new technology and a startup. And all this are transforming the way we approach healthcare, diagnostic, and research. In our conference, we have two parts. In this first part, we will explore together how medical startups and innovative applications are shaping the future of healthcare. I will start by introducing the concept of medical startup and the concept of medical application before handing over my colleague. My colleague will present to you one application named ChronoLab. ChronoLab is a medical application we have developed to address the challenges of modern medicine. Let's go. The medical startups are at the forefront of medical innovation, introducing the solutions And the solution enhance the patient care and optimize the medical workflows and improve their accessibility. By leveraging the digital health technology, the startups are transforming the healthcare landscape. While some view them as competition, many established healthcare firms recognize their potential and try to collaborate with them in order to accelerate the digital transformation. So we can say that the startups bring a fresh vision of modern healthcare, making it more efficient and tailored the need of tech savvy and also independent patient. Our objective is to understand what medical startups are and their key development stage. This is very important for you. If you want to create a medical startup, you must learn the key development stage. Here we have 7 stages. Then we compare the medical application to the medical startups, and we identify the challenges and opportunity in the health tech sector. Then my colleague will examine ChronoLab as a case study of a successful medical application. Now we define what is the medical application and what is the medical startups. The medical applications are digital tools. These digital tools designed to assess the healthcare professionals and the patient in order to give information for the diagnosis, for the treatment, and care monitoring. However, the medical startup is innovative company and not a digital tools. These innovative companies leveraging the technology to develop a new solution for the diagnosis, the treatment, the patient management, and the healthcare efficiency. This is 2 tables which compare the medical application and medical startups. This is the similarity and this is the difference. For instance, the medical application improve the access to healthcare and treatment efficiency, and the medical startup create a sustainable solution to optimize medical care. So probably they have the same objective, the improvement of healthcare and treatment efficiency for the medical application and the optimizing of medical care for the medical startup. So for the difference, as I defined the medical application, it is considered as a digital tools for healthcare support. However, the medical startup is defined as innovative company. So this company is developing innovative healthcare solution. Also, for the user, here we have a difference. The user of medical application are the patient, the healthcare professional as the doctor. However, in the case of medical startup, we have this healthcare professional and the investor and hospital, in addition to the patient and the doctor. This is the difference between the medical startup and the application. I gave you some example of the most famous medical application and most famous medical startup. For the medical application, we have MediSafe. This medical application helps patients manage their medication intake with the reminder of personalized treatment. The second medical application is ADA Health. Here, we have the use of artificial intelligence. It is artificial intelligence powered application that will provide the preliminary symptom assessments. And the last one is Doximity. This Doximity is a secure communication platform. So it is a communication platform for the health professional. Now we pass to the most famous medical startups. The most famous one is Medtronic. It is considered as pioneer in development of a pacemaker and other medical devices. We have Genentech. This medical startup is specialized in biotechnology and innovative therapy. And the last one is Babylon Health. This a company offers remote medical consultation through artificial intelligence and telemedicine. So you pose the question, what is the correlation between the two? So we can say that the medical application is a tool or is instrument which is used by the medical startup. The medical application, as I said before, facilitate the diagnostic, the treatment, and the patient monitoring through the digital tools. However, the medical startup developed innovative solution integrating the technology into healthcare, and this company can use the medical application, both improve the access to care, optimize the patient management, and promote more personalized medicine. Now we pass to the evolution of medical startups. This is the historical background. The rise of medical startups began in the 20th century with the gradual integration of the technology into the medicine. The first company is Medtronic, which is founded in 1944. This company marked a turning point by developing the first battery-powered external pacemaker in 1957. The second most famous startup is Genentech. This company is considered as a pioneer in biotechnology. Why this? Because it created a revolution in the diabetes treatment by the production of synthetic insulin in 1976. This Genentech create or product a synthetic insulin. This is why this medical startup, create a revolution in the diabetes treatment. So, these innovations led to the foundation for more modern medicine and encouraging the emergence of startups which focused on telemedicine, artificial intelligence, and biotechnology. We pose question, how entrepreneurship is shaping the future of healthcare. Here we have these three ideas. So the first one is bridging inequality by improving the access to healthcare and reducing the cost for more equitable patient care. The second one is the driving disruptive innovation by introducing new approach in the diagnostic, treatment, and patient engagement. And last one is the optimizing the hospital efficiency, and this by using the automation and artificial intelligence. These two the instruments can enhance the team coordination and reduce error and free up time for the better patient support. This is the best information for you. When you want to create a medical startup, what are the key stages? Here you have a 7 stage. You must learn this 7 stage. The first one is the creation or identification of the idea and problem. Here you define an unmet medical need and propose innovative solution. Then you analyze the demand, the trend, and gather feedback from the professional and the patient in order to have the validation of your idea. Then, the business model and strategy development by developing viable business model and strategic roadmap. Here, the fourth stage is the product development and regulatory approval. Here, you design the solution, you obtain approval, and ensure the compliance with the regulation. And here, this step is very important for you because we will have some challenges, the funding and investment. So you secure the funding from investor and grant and other financial sources. Then you have the commercialization and the scaling of your product. Launch your product, acquire customer, and optimize the expansion. And the last one is the sustainability and the growth. Here, you ensure the profitability and you foster continuous innovation and anticipate industry changes. This is the startup life cycle. Here we have these steps. So we pose a question: what are the challenges faced by the creation of medical startup? Here we have these challenges: the regulatory and compliance issue. Here, the strict healthcare regulation and approval, and with a high cost and a longer time for the compliance are considered as the first challenges. We have another challenges, which is the funding and financial sustainability. We will have difficulty in securing investment, and this is due to the high risk nature, and we need for sustainable revenue models. The third challenges is the technological barrier and adopters challenges. The integration with existing healthcare infrastructure is very difficult. And the most famous challenges is the resistance from the medical professional and institution. And here we have the participation of the market competition and entry barrier. Some market competition forbid the creation of new medical startup. So this large established company dominates the market, and the high cost of marketing and customer acquisition is very difficult for you. So for you, in order to overcome the challenges in the healthcare technology adoption, you must have this information. The most famous challenges are the lack of alignments between the technology and healthcare goals, the lack of user involvement and feedback, and the lack of resource and support. These three challenges face every person who will create a medical startup. So you must have idea about the most famous challenges in healthcare technology adoption. So you pose question: what is the future of medical startup? The future are the emerging trend in healthcare innovation, here by the use of artificial intelligence in diagnostic and treatment, internet medical things for real-time patient and monitoring, real-time patient monitoring, and also the big data analytic for predictive healthcare, and also the blockchain for securing medical records and the use of telemedicine and remote care advancements. Also, the artificial intelligence play a great role in the giving information for the diagnosing. So it can reduce the human error. And the robotic-assisted surgery can improve more precision than the human surgery. And the virtual health assistant enhance the patient engagement. This is the role of artificial intelligence and automation. For the investment and growth opportunity, here, the increased venture capital in health tech with the collaboration between the startup and hospital can consider it as a form of growth opportunity. And also, all this in order to have a good expansion of digital health ecosystem. You pose the question: can you give me the prediction for the next decade? So for me, according to the literature review, the prediction for the next decade are a personalized medicine, which became mainstream, and the artificial intelligence driving drug discovery accelerate the treatment, and we will have the integration of augmented reality in medical training. This is the future of the medical startup. Also, this is very important for you. You should know the legal and ethical consideration. We have the data privacy and security, the patient consent and trust, liability issue, and ethical consent. For the data privacy and security, here, Handling the sensitive patient data requires adherence to some laws like HIPAA, and all this in order to ensure the confidentiality and cybersecurity. This is in order to have a security of information. For the patient consent and trust, here you ensure the informed consent for data use and you maintain the patient trust in digital healthcare solution. For the liability issue, we can pose a question. Who is responsible when a digital application or artificial intelligence provide incorrect diagnosis or medical recommendation? Is it the medical application or the person who use this medical application? This create a kind of liability issue. For the ethical concern, the artificial intelligence-driven diagnostic, also telemedicine and automated decision, all this raise ethical question regarding bias, transparency, and the patient autonomy. So you must learn this legal and ethical consideration before creating your medical startup. I gave you some recommended book and resource. The first one is the book for understanding the medical innovation and artificial intelligence. Eric Topol have two books. The first one is the title is The Patient Will See You. In this book, he explained how digital medicine is transforming healthcare. In the second book, with the title of Deep Medicine, he showed how artificial intelligence can make healthcare human again. So he showed the impact of artificial intelligence on the patient care. The last book is The Future of the Medicine, the Technology and the Human Touch. Here, the author explores the future medical trend. We have also we have this other books for the startup and entrepreneurial success. Here we have the first book, The Lean Startup. This is very important for you. Why this? Because the author gave you the methodology for startup success. The second one, 0 to 1. Here we have the building innovative business. So it's concerned the businesses. And the last one is the most important one. It is considered as the guide for startup growth. This book title is The Startup Owner Manual. It's a manual. It is considered as a guide for startup growth. So you can read this book, which is very, very important for the startup growth. We have also online research and journal. We have the Harvard Business Review and Med Technology Review. So for the first review, it shows you some case study on startup. And the second one is cutting-edge health tech trends. So for you, the key takeaways are the first one, the medical startup and innovation are transforming healthcare accessibility, efficiency, and quality. Also, the digital application like ChronoLab play a crucial role in optimizing patient management. The overcoming challenges such as funding and regulatory compliance is essential for your success. And the collaboration between the medical professional and tech entrepreneur drive a meaningful innovation. And for the future, in the future, we can have an increased use of artificial intelligence, blockchain, and other digital tools in the healthcare. And we will have also the expansion of telemedicine and remote patient monitoring. And we will have the personalized medicine, and this is through a big data and predictive analysis. Now that we have explored the first part of this conference, I will now hand over my colleague who will present to you this case study of ChronoLab. So, this ChronoLab is a kind of a medical application. She will explain in detail that what ChronoLab is, the key feature and functionality, the impact on healthcare, and its implementation process. I invite you to listen carefully in order to gain a better understanding of how the innovation contribute to the evolution of medical practice.
Good morning. I am Assia. I am a doctoral student in Polytechnic School in Oran. I am here today for presentation of the ChronoLab, the medical application of the future, is smart, efficient, and connected. This application of the future that optimizes analysis management and improves the patient experience. The problem is the medical analysis laboratory play an essential role in the diagnostic and monitoring of disease. 20% of patients doubt the reliability of their test results, raising concern about the quality of laboratory equipment and practices. And 40% of respondents report long wait time for their test results, which can delay diagnostics and treatment. ChronoLab, our solution application, optimizes analytic management and approves the patient experience. Each patient is unique, which is why the ChronoLab adapts to your specific needs to offer you an optimal experience. The ChronoLab's mission is to simplify the care pathway from making an appointment to transmitting results to preparing for analysis. And our business is to become the leader in smart solution by offering innovative solutions to redefine the standard of medical analysis. For the doctor, will be able to draw up a list of tests to be carried out and communicate it to the patient so that he can make an appointment. Or the patient will consult the detailed list of analyses to be carried out, make an appointment with the laboratory of his choice, or the doctor will do so. Then they will be able to prepare themselves thanks to our chatbots. The laboratory will receive appointment requests in a dematerialized manner in order to plan and validate them. The laboratory will send a notification to the patient. The result will be sent to the patient and the doctor. How do it work? The doctor orders tests. He charts them with his patient and laboratory. The patient makes an appointment with the laboratory or the doctor does it. The laboratory confirms the appointment and sends the patient to the notification to the patient. The laboratory sends the result to the patient and the doctor. Finally, the doctor sends his report to the patient and gives him a new appointment. Let's now test the application. The application is here. You can sign up with our Gmail account, Microsoft account, or application account. There is the interface of the application. You have got a chatbot here and another chatbot for the patient if you want to prepare the medical analysis. The chatbot of the Chronic Labs analysis is here. For example, you want to do a lipid test. You ask the chatbot how to do a lipid test. Lipid test. There is the answer for the preparation of the lipid test. You have got another question. What are normal lipid levels? How often should I get tested? What effect lipid test result? Can I eat before the test? For example, can I eat before the test? There is the repeats. Exception for healthcare providers may allow not fasting lipid test, but it is now less common. So there is the chatbot for preparing of analysis, medical analysis. Then, if you click on Doctor tab, you would be directed for two forms to register a new patient, to make a note of doctor, to make a report of doctor, and the listing of the analysis. For example, I am click formula of analysis. The doctor can receive the listing of analysis, and you can test the listing and save, print, or submit the listing of analysis. How to make. Our advantage: significant time saving, significant improvement in accuracy of results, full compliance with current regulation, and substantial cost reduction. Our SWOT analysis is the improved efficacy, accuracy time saving, regulatory compliance, and flexibility. The growing market is the customization of solution, integrations with other technology, and provided collaboration. Opportunity is high initial investment cost. Is not opportunity, is the fabulous high initial investment cost, complexity of implementation, technological dependence, and resistance to change. Increases competition, rapid technology change, and cybersecurity, regulatory resistance. For conclusion, the Chronic Labs is a first step toward the medical laboratory of the future, where technology and the service of health makes it possible to improve patient care and facilitate the work of professionals. We are convinced that this solution will help revolution the healthcare sector. Thank you for your attention.
Thank you very much, Assia. We thank Assia ALLAL and Dr. HABCHI Nawel for the recording. I think Dr. HABCHI Nawel is also in the room today. Is that correct?
Yes, I am
will-
here.
Hello, Dr. HABCHI Nawel. Welcome. Thank you for the speech.
Thank you very much. Hello,
I was
everybody.
interested with your AI technology.
Yes. So I usually present my content live, but due to a personal commitment, today I have recorded my presentation for this session. You will be able to follow it again.
Yes, that's great.
So,
That's much better actually, because normally live presentations can go a little astray because of excitement and so on.
I want to present in the first section as a courses of how to create a medical startup, the the 7 stages. I want to present the 7 stages and also the challenges that we'll face in the future. This is for the first section, and in the second section, my colleague present to you the Chronolab as a case study of medical application, and she gave you information step by steps. This is an example of the use of medical application. If you have a question, you can write.
Yes, I have a question. Do you think with this advanced technology, we can also decrease the amounts of animal experimentation?
Yes, for the experimentation, it is not easy, especially in the African country. We have the problem of inflexibility between the doctors, the patient, and the laboratory.
Okay, there's one more question. With the rise of AI-driven medical applications like Chronolab yourself, how do you see the balance between automation and human expertise evolving in the future healthcare systems? Is
So
that going to be making it easy for laboratory assistants and researchers?
So for this question, you pose a question, the difference, the balance between the automation and the human expertise in the future. This is your question.
Yep.
Yes. So the balance between the automation and the human expertise in the future healthcare system will likely evolve into the complementary model where the artificial intelligence enhance rather than replace the human decision making. So this artificial intelligence-driven application, like our cases of Chronolab, can streamline the diagnosis and personalize the treatment, and also optimize the workflow efficiency and reduce also the physician workload and improving the patient outcomes. But the human expertise remains crucial for complex decision making and ethical consideration. I show to you the ethical consideration that should make in consideration the ethical, the patient consent, the liable decision, and also the patient interaction. So the key aspect of this evolving balance can include the augmented decision making, the precision of medicine, and also the ethical and legal oversights. This is my answer.
Okay, thank you very much. It looks very promising for the very hardworking laboratory researchers, because I know that they're going through very, very difficult times with their hard workload, and it is very promising for them. I'm happy for their behalf with your advanced technology and applications, Dr. Nawel HABCHI.
Thank you very much.
You're welcome. So, is there any more questions to Dr. Nawel HABCHI and Assia ALLAL? Please type in the chat box if you have any. Okay, looks like there isn't anymore. Is there anything else you'd like to add, Dr. Nawel HABCHI, to our audience? Okay, there's nothing else to add. So the last speaker is me, myself. I'm introducing myself to you. I'm Pai Sengul. Everyone calls me Pai. I have another name which is very hard to pronounce. It's Pinar, but because of my education being done in Germany and in England, nobody could pronounce the name, so I did change it to Pai eventually, and it stayed like that. Today, I'm going to make a very interesting presentation to you. So I will start if you guys are ready. Please feel free to add any questions in the chat box, or you can interrupt with your microphone as well. Can somebody just approve that they can see my slides? Can someone just tell like they can see the slide or not? Anyone? Anyone courageous enough to say something? Please? Nobody. Okay, I see the chat box. Okay. Can you also see if it moves though? Can you also take the microphone because yeah. Do you see when I'm moving the slides? Can you see? You can just unmute yourself. Okay. I just before starting, I just would like to encourage you guys to take the microphone more often, because in life you need to really be a little more extroverted to take parts in things. So please do not always be retreated to yourself, because we need each other and we are social animals, right? Okay, today, good evening everyone, and good afternoon to the people that are listening from other parts of the world, like Europe, etc. It's a profound honor to be here today, not just as a neuroscientist, but as someone who believes true scientific progress is measured not only in data, but in the depth of our conscience. Today, we gather here under the vital banner of the brain health, mental well-being, and neurological innovation. Yet today, I ask you to look beyond the surface, to examine the very foundation upon which our methods are built. And I'm here to challenge a persistent illusion in biomedical science, unfortunately. The outdated notion that we must inflict harm to achieve healing, that progress demands the suffering of another living being. Here in this picture, it's an allegorical image of animals doing experiments on a human being rather violently, because we can see blood and it must be hurtful, wouldn't it? Because if there is blood, the skin barrier is hurt and also the blood vesicles are hurt, so the blood is coming out, right? So there is pain. We can empathize with it because we are humans, and there's a fellow human being in front of us in the picture, lie down, also bands around the arms and the legs, so they cannot consciously go against it. They're out of their wills to get out of that table, and under the scissors and all kinds of knives using by other animals, non-human animals. So this here, even though it's a cartoon, we can empathize as a human being. We don't want to be there, right? We don't want to be there. We don't want to be under the scissors. And it's another human being with a very similar system to us. Brain, head, arms, legs. So we can empathize. We can use our mirror neurons here. This is why I start with this image. So as people mature up, it's a developmental stage in developmental psychology. We learn from Erikson, Piaget, and many other famous psychologists that when you're an infant or a child, you are egocentric. So it says that when you're a baby or a child, you tend to oversee other people's pains. You are all about yourself. But as you mature up, you can resonate with others. You can see their pain. You can empathize. Empathy comes with maturation. And I invite all of you to put your empathy scissors, glasses, and be more empathetic during this presentation that I'm going to present to you. And I believe that all of you are at least university students, undergrads, postgrads, or even PhD candidates, and even like academicians. So you are able to empathize with your cortical maturation process nearly being developed or already developed. So I start with a quote with Mahatma Gandhi, as we have a lot of audience from India. They would, I think they like Mahatma Gandhi. I know that they like Gandhi a lot. So the greatness of a nation and its moral progress can be judged by the way its animals are treated. So Gandhi says in here, nation, we will replace it with scientific, let's say, scientists under the umbrella of science. The greatness of scientists can be and their moral progression can be judged by the way the animals are treated as well. Let's ground this discussion in undeniable facts, in hard data, not mere sentiment, okay? I was talking about sentiments. Now I will go to data so people with mathematical minds can also take a nice breath. According to the US FDA data in 2021, a staggering 90% of more of drugs that demonstrate promise in animals trials ultimately fail when tested in humans. I am repeating myself, 90% of animal experiments, when they are transformed to humans, they fail. It's 90%. It is not trivial, is it? So this isn't a minor imperfection. It's a systemic flaw with devastating consequences. Vivisection, by the way, it means from Latin, vivi means alive, and section means cutting in pieces. So vivisection is cutting animals in pieces in very raw terms. So we tend to use Latin, sometimes avoiding the realities. We suppress our feelings. In Freudian terms, we repress our empathy, we repress our conscience and our thoughts as well. So vivisection, cutting animals in pieces, remains a standard despite scientific and huge ethical flaws. Consider, for example, the fight against Alzheimer's. Over 99.6% of the treatments that showed initial hope in animal models crumbled in human trials. 99.6% animal models that did treat Alzheimer's in rodents could not treat Alzheimer's in humans. Like, there is only 0.4% of success. And if you had statistics during your undergrad, you would know that 0.4 is not statistically valid, right? You need to have 0.5% of error rate. Here it's the vice versa. So why? Because of the intricate complexity of the human brain and its diseases. It often cannot be accurately replicated in animal systems. In the realm of stroke research, the same goes on as well. One successfully translated to human patients only about from 1,000 potential treatments that proved successful in rodents in stroke, only one was successful. So 1 in 1,000. What is that? 1 in 1,000. 0.001% of success, right? It is, as any statistician wouldn't approve that as valid, right? It will not reject the H0 hypothesis in statistics. It will not. The p-value is laying low, it's under the ground, and the effect size is nothing, obviously. If you have a very low p-value, the effect size cannot be high as well. So as one more disease that I would like to mention is Parkinson's disease. And the commonly utilized MPTP lesion monkey models, they also fall very short of representing the full spectrum of human disease pathology. So Alzheimer's, Parkinson, stroke, and also MS, I will also talk about that. They always fall short in treating humans. Yes, they can treat non-human animals, but not the human animal. So I would also like to give a little briefing about MS because I'm working on MS, multiple sclerosis. In animals, MS is widely used and tested on animals. And it's a very funny thing because did you know that when you trigger inflammation or degeneration like MS in animals, or mostly it's rodents, by the way, they don't really get MS. It's not the same. So they call it something else. It's called experimental autoimmune encephalitis, EAE. So most people, most biomedical scientists, biologists, molecular biologists, geneticists, even neuroscientists that work on multiple sclerosis will tell you that they are finding a new drug on MS that is very efficient. And yet you ask them on which materials did they work with, what were their methodology, their participants, they just tell you that they had lab-grown rodents and they degenerated their brains using toxic chemicals. And in the rodents, we don't see many facets of what we call MS. MS is a cognitive degeneration. Comorbid with the problems that they have, like with the motor function problems. So they have fatigue, they have brain fogness, and in rodents there isn't any. Also the immunology behind MS tells us that it's B cell driven, whereas in rodents it's T cell driven. It's a complete another cell. Obviously they're both white cells, but they're a huge different subtype. When you are working on molecular biology or immunology, you know that they have very different functions when it comes to T cells and B cells. And in experimental autoimmune encephalitis in rodents, they see the lesions only in spinal cord of the animal, whereas in humans, we have it all around. It's mostly in the brain as well. Like, yes, they can have spinal cord lesions, but they have it all around the brain too. And according to the McDonald criteria, which is the guideline for diagnosing MS, you have to have at least, let's say, 2 spinal cord lesions and 1 brain lesion. It's mostly there's always a lesion in the brain most of the time. So in EAA, in the animal model, there is no brain lesion, and MS is a neurodegenerative neuroinflammatory disease which needs brain, and there is no brain lesion in the animal. So how can we really compare that? I already talked about the cognitive function change. There is no cognitive disorders in the rodents that have MS, let's say, MS mimicking. And also people with neurologists that work with MS would know that there are subtypes of MS, like primary progressive MS, secondary progressive MS, relapsing remitting MS, etc. And in the animal model, there is no subtype. So all of the mimic disease are the same, but in humans, some of them get it at the age of 20 and it slowly progresses, and maybe they get it at the age of 50 and they progress very, very fastly, and they can end up even dying within a few years. So there's a huge change and variation within the this disease as well. So there is a huge neglect when they're working with animal models, telling public that they're actually working with that disease. That disease is not the same when you transfer it to the animal. You have to understand. You have to really admit that. They said, okay, experimentally, autoimmune encephalitis, an animal model mimicking MS. It is trying to mimic MS. It is nothing near MS. It just has some similarities, and don't most diseases have similarities? It takes a huge time to differentiate which actual disease you have. Even with very simple diseases like the flu, you can have adenovirus, you can have rhinovirus, you can have influenza virus, you can have the pig virus, like pig or the chicken pox, or like it can go to 100 other alternatives, right? So you have, they all have fever, most of them have runny nose, you can have a cough, like COVID. I forgot about COVID. It's the most popular one lately. Anyways, like you can have fever, runny nose, cough, and you cannot still know which virus you still have or bacteria. So having similar symptoms is not enough for diagnosing your disease, is it? So please remember that. And animal models mimicking any disease is not enough to assure me or make me convinced that they can treat that disease that's found in humans. So this is what I've already mentioned to you. You can have a look again. You can see that 99.6 fails in Alzheimer's. One in only in 1,000 works in stroke. In Parkinson's, the same. So the conclusion, we are investing billions into a method that does not work. So what is the millions? I'll tell you about that too now. In 2019, you can see the caption below, the global animal testing market was significantly larger at $10.74 billion compared to the global non-animal alternative testing market at $1.11 billion. So you can see there's nearly 10 times difference, right? 10 times difference. 10 times you pay less, 10 times cheaper, and you get even better results. Better, easier, cheaper, and what about the ethical part? No pain, without pain. Ethical, cheaper, easier, ethical, and better results. Why do we still persist on doing animal research? That's my question. So we are investing vast resources, billions of dollars into a methodology that not only frequently fails to deliver, but often misleads our research efforts. It misleads. It's loss of effort, loss of time, loss of thousands of hours working in the lab for human fellow researchers as well. Remember that you're losing your time for no good cause. You're not gonna find the effective drugs if you want to find it. You're doing so much bad with being persistent on doing animal experimentation. And if you're doing something like that, it's not late to change your experiments. You can change your supervisor. If you're doing a PhD with a supervisor that does animal experiments, find another one. The world is full of supervisors. You can find a new one. You can find a person that works with humans, that works in silico, that works in vitro. That's what I've done. I had a challenge as a neuroscientist finding somebody that did not work with animals, but at the end I did in my university. I did find a very successful supervisor who works in biochemistry, in medical biochemistry, and he was very, very keen to working with humans. He was already working with human participants for many diseases. So you also can, okay? You just shouldn't give up the challenge, and you have to be a little more extroverted. Take the mic when you need to, guys, okay? Okay, so the true scientific advancement isn't about blindly following habits. It's about courageous questioning and revolutionary thinking. Beyond efficacy, now I'm turning towards more philosophical ground. As I've done a philosophy BA as well, I would like to talk about ethics. Ethics is the philosophy of morality, for those who doesn't know. ethics, philosophy of morality, okay? So ethics can be divided into two. It can be applied ethics and the classical ethics. Now I'm going to talk more about the applied ethics, and I would like to convey my regards to Professor Dr. Peter Singer, whom I really love very much. He's an idol for me, and I have translated one of his books into Turkish because I'm Turkish. And I will just show you that, just a moment. So this is Peter Singer's book. I will just stop sharing so you can see better. Oops. Back to meeting. Stop share. Okay. So this is his book. Why Vegan? This is the Turkish version though. And you can see my name there. Where? Oh, here. So this is a very tiny book called Why Vegan, but I do advise everyone to get this book. Also, there is another book by him called The Buddhist's Dilemma. I think most Indians would be very interested with that, if I'm not generalizing, but you have to have a look at that as well. Peter Singer. He's a professor of philosophy and doing research, has been writing loads of literature on veganism, ethical veganism, animal rights activism as well. So I'm going back to my presentation. So I'll talk about the ethical imperative right now. Let's turn to crucial realms of ethics. It's not as an abstract philosophical debate, but it's a matter of applied morality. And in our scientific endeavors, this practice of vivisection is not simply inefficient, but it is inherently violent. And I'm calling you to be in communication with your hearts or your conscience, to be more specific. You have to find that connection again, because I see that we all lose our connections to our consciences and our hearts during PhDs, masters, and higher education. And it is not a must. We can still hear it. If you're working with animals and if you're hurting them and if they're making a voice, if you can feel that they're being hurt, just listen to your heart and leave it at that point. You can leave. You can leave. You're somebody that has gotten into the stage of PhD. You can find a new supervisor. You can find a new research. It's never too late, okay? It's never too late. Please do whatever you're doing with your hearts. Please do not be hypocrites to your hearts. Listen to your heart. You matter more. Your heart matters. What you feel matters. Stop suppressing your feelings. Okay, enough activism there. So, mice, rats, and primates, these are not mere biological tools. They're sentient beings capable of feeling stress, happiness, forming bonds, and responding to trauma in ways strikingly similar to us. To subject a monkey to paralysis, to deliberately introduce a disease, or into a vulnerable puppy, or to invasively open the skull of a mouse for data that will likely prove irrelevant. This is not sound science. It is cruelty cloaked in the guise of a laboratory procedure. It is cruelty cloaked in the guise of laboratory procedure, guys. A truly evolved science must be both rigorously accurate and profoundly compassionate. So there is a horizon of hope. Do not be hopeless. There are effective and ethical alternatives. So is there anything I need to mention from this slide? I said mice, monkey, dogs, they're all capable of pain, fear, and suffering. And why? Just telling you very simply, they also have a CNS, central nervous system, which the pain receptors, nociceptors, they're called, are also found in them. Okay, they're also present in them. So they also feel what we feel. When you push when you hit your toe into a table, it hurts. The same happens with them, okay? So I'm calling you guys, all of you, to replace outdated harm with conscious innovation. I'll tell you how we can do the innovation, but if you're a researcher, you already know there are alternatives. You already are knowing that there are alternatives. Let me introduce them to you if you don't know about them. But before that, let's see this image, why we should go to alternatives. Look at the monkey. Look at the monkey in pain, right? There are blood around. There is a whatever it is. There's a wooden something. There's a plastic. There's a road in the skull. Just think of you being hit in your skull, just being hit. Can you imagine having a rod inside of your skull? You don't want to imagine, right? You will not let anybody do that to you consciously if you have your willpower and if you have a free will, but they don't have a free will. Nobody gotten their conscious approval. There is no ethical approval, right? So it is not just done in primates, because primates are being restricted mostly in some of the like in the US and the EU, they have strict guidelines for them, but they're still being experimented overall in other words, in other parts of the world. And the most common ones are on the right hand side. You can see that there's a rat. They are the biggest victims of experiments that we are doing to them non-stop, and also followed by the rabbits, followed by dogs, and then the monkeys. The monkeys look alike to us a lot. We can really resonate with them. We can empathize with them because they have similar configuration of even the face, right? Eyes here, eyes in the middle, unlike rabbits on the side. So you can empathize with the monkey. Why not empathize with the puppy? Why not empathize with the rabbit? Why not empathize with the rats? Empathize. You could even be born as a rat. You could be that rat. Just empathize, please. If you're a human, you have higher cognitive skills. So use it for the goodness of the world. So the horizon hypothesis, the empowering truth is that we no longer face a first choice between scientific progress and ethical conduct. A wealth of innovative alternatives is emerging, offering more human relevance and compassionate pathways to discovery. So the first one is organ on a chip. And then brain organoids and in silico simulations, microdosing imaging, and microdosing plus good imaging. Just a moment. And iPSC technology. iPSC is the induced stem cell, pluripotent stem cell technology. Sorry, I need some light because in Istanbul it's getting darker, but it's not working at the moment. Just a moment, I'll take a moment. Sorry for the interruption. Organ on chip technology, I'll tell you very, very easily for you so you can understand. Organ on chip is a tiny, you think of tiny plastic organs with living human cells. So they're on the little like USB sticks that are transparent, and you can see that tiny living organs in it. So they're called organs on a chip, or OLCs. Think of it like The Sims, the game for organs, okay? So the brain organoids, they are human stem cell derived neural models. What does that mean? For ease of understanding, they are mini brains from stem cells in a dish, okay? And they're used mostly for epilepsy. I'll show you the picture for that. This is brain organoids. They're used for epilepsy, autism, and Alzheimer's disease. Yeah, the negative side might be like the notion that they're not conscious. they are not living beings, right? So they're not conscious like us, but they are complex enough to show brain-like behavior. And you can see here the organ on a chip. This is how the organ on a chip is done. You get some cells from these specific organs and then put them on the chip and give them enough the needed environment and then grow the organs, all right? And these are mostly used for testing drugs. If you're working with drugs, you can test drugs on organs on chips. And if you're working with epilepsy, autism, Alzheimer's disease, here are your brain organoids. And in my university, there. Okay, great. So there is somebody in our university doing organoid labs. They have brain organoids laboratory going on. So, by the way, I would like to mention when I'm talking about, for example, organs on chips, right? Where is it? Here. You have a question in your mind. Is that possible or is that very common in the world? I have answered that question for you. I did my research. So in the US, it's 45%. So 45% they are used. It's a very high percentage. And the biggest company doing that is called like the funding body is called Emulate, and also it's done in the Harvard Vis Institute. Other than the States, these labs are found in Europe, especially in Germany and Netherlands, about 25 to 30%. And the biggest funding bodies are the Horizon Project, ORCID, and EuroOC, followed by China with 15%, then Japan with 5%, South Korea with 2 to 3%, and rest of the world with 5 to 8%. In the rest of the world, the growing interest is in India, thanks to all of those Indians doing that work, and Brazil, Israel, and Australia. When it comes to other alternatives, I mentioned these two, and the in silico simulations. In silico means through the brain, through the computer, all right? In vitro, it means, in vivo means using an animal, living being. In silico means the computer, and in vitro means using a glass tube, all right? So in silico inside a computer, this is an image for that. These are used mostly for saving years of lab work because they can do everything like very fast. It's like what we are using right now. We are using artificial intelligence for all of our works, right? So this is like using artificial intelligence in the lab itself. Great. It's great innovation. And I think Dr. HABCHI Nawel also talked about that during her presentation with Chronolab. So it's something like that. In silico is simply computer brains testing drugs, virtual models of human cells, organs, or even whole body systems. They can mimic them. They can make research on whole body systems as well. And this is like AI plus the real patient data. You have to upload all of the patient data, and then it makes statistics out of that. Also, I do something like that using my advanced statistic methods, using random forest. So when you have, let's say, 5 participants in your research and they're not enough, you can idealize that, repeat that experiment, let's say 1,000 times more, and see if the experiment is good enough. And I accidentally raised my hand to myself. And there is microdosing plus imaging. This is a very easy method, even without knowing brain organoids, organ on a chip in silico, because these are very advanced technologies. All of them are advanced, but microdosing is not that advanced. Okay, imaging is advanced, but we could have done microdosing even in the years of 1950, 1920. Microdosing is very easy. You just take, for example, 100 micrograms, but you divide it into, let's say, 10 parts and give that little bit of that dose into the human. So this is on humans. This can be also good and bad. You can have ethical considerations for humans too. So that's good if you have ethical considerations. So why not replace it to animals there? I'm getting tired with my presentation being so long, so I'm gonna cut it very fast. So please stick with me. So microdosing plus PET and MRI. So microdose, give a little bit of medicine to somebody and then image them using high imaging techniques like PET CT or PET MRI. All right. So these are safe and precise. And lastly, okay, let's have this is an image, microdosing. Little, very tiny medicine, very tiny drug injected to the human or orally taken. And then they will be imaged, brain imaging or whatever. So the last alternative is the iPSC technology, which is induced pluripotent stem cells. It turns a skin cell into any cell you want. They take a cell from your skin and then they can turn it into heart cells, neurons, liver, or anything you can imagine. So these are here, yes. So before, is there anything else I need to mention? So these are the domains that we are ready to transform: pharmacology, toxicology, neuroscience, psychiatry, education. In neuroscience, I'll mention we use human-derived brain organoids. They are the best ways to mimic human brain. In pharmacology, you can replace also with organoids and AI. In toxicology, in vitro is still good. In psychiatry, digital biomarkers, and I think in silico can be helpful as well. I'll talk about the emotional intelligence. So the underlying concern that most of the researchers will be if we move away from animal models, do we somehow lose control over our research? I argue the opposite, because by embracing more human-relevant methodologies, we gain a more precise and ultimately more effective understanding of human health. Progress built upon a foundation of harm is inherently fragile, destined to crumble under the weight of its limitations. To be a scientist in 2025 demands more than just a deep understanding of the hippocampus or the synapse. It requires cultivating the emotional intelligence to critically evaluate our methods and to declare with conviction there is a better way. We can achieve progress without inflicting suffering. So progress doesn't come from perfecting the past. It comes from letting go of it. We have been trying to perfect the past with animal experiments. It is our past. There is no way of changing it, but we can change the future. So we stand at the cusp of a new era where artificial intelligence can stimulate complexities and our understanding of consciousness extends beyond the human realm. In the transformative time, let us not be encumbered by outdated paradigms. Let us build a future of science that reflects the very best of our humanity, not just our intellectual capacity, but our unwavering integrity. Science that does not breed suffering, laboratories that do not silence screams, data born of empathy, not exploitation. This is my vision of ethical science. It should be yours too, please. This is not utopia. This is already happening and we only need to choose it. I just mentioned to you that in the States, 45% of the labs are already using organs on a chip. Why not you? Why not you should be the revolutionary one? So, a science that chooses not to silence the cries of vulnerable beings in the pursuit of healing, but instead of amplifying the voice of our collective conscience, in the unwavering pursuit of the truth, because the feature of brain health and all kinds of health, and indeed all of the medical science, will not just be free from animal exploitation, it will be wiser, it will be more just, and ultimately it will be more beautiful. Thank you for listening, and I'm open to any questions from the microphone. You can just use your microphone, please. Open to any questions. Thanks for listening. And we are expecting your registration for the upcoming OLCIAS International Live Conference. You can find it on the website. Any more questions will be welcome. Thank you. Oh, okay, Gaval. This is from Gaval. Thank you. Thank you. Thank you for putting a smile in my face, Gaval. Namaste. All right. Hope you guys read more, do more science, do more research, improve yourselves, and do not hurt animals. Stay well. Goodbye, everyone.